네, 안녕하십니까. 대한수학회 대중강연을 시작하기에 앞서 우선 유튜브로 참석해주신 여러분께 감사의 인사를 드립니다. 저는 이번 대중강연 좌장을 맡은 카이스트 수리과학과 그리고 IBS 의생명수학그룹의 김재경이라고 합니다. 지금부터는 영어로 강연을 진행할 수 있도록 하겠습니다. 먼저 오늘 연사분을 소개해드리겠습니다. I have the great pleasure of introducing Dr. Jeremy Gunawardena as today's public lecture of Korea Math Society annual conference. He got a PhD for algebraic topology from Cambridge University. Then he suddenly switched to the biology field, and he became a faculty at systems biology at Harvard University. And he was also director of virtual cell program at Harvard Medical School. And Dr. Guenardera has used various mathematics theory and tools to investigate cellular signaling system. And he published many prestigious journals, including Nature, Cell, and Nature Cell Biology. In fact, when I was a student, I also read his paper a lot. So today, he will talk about topology and gene, which looks very interesting title. So please join me welcoming Dr. Guenardena.
Jay, thank you very much for the introduction. It's a pleasure to be here. Let me start by saying thank you to the Korean Mathematical Society and also to you, Jay, for the honor of this invitation. I regret that I'm unable to be in Korea to present it in person. I had other commitments at the time, so I'm actually in Barcelona, Spain, where I'm visiting for a little while. I'm most grateful to my host here, Jordi Garcia Ojavo, for loaning me his office. So the background that you see behind me is borrowed for the purposes of this presentation. So thank you all for joining us. Let's start with some things which I'm sure will be familiar to everyone in the audience. This is showing Jim Watson and Francis Crick, the discoverers of the structure of the molecule deoxyribonucleic acid, better known as DNA. It was taken, I think, not long after the structure was discovered. And I'm sure you will also be familiar with the structure itself, which is visible so often these days. It is a particularly beautiful structure, and many chemists and biologists would regard it as the most beautiful of all molecules. It consists of two strands which are linked together in this spiral shape of a double helix. And each strand, as I'm sure you know, is a sequence of chemical so-called bases. There are 4 of them: adenine, thymine, cytosine, and guanine. And each strand is just a sequence of these 4 bases, and the 2 strands are linked together through quite weak hydrogen bonds in such a way that adenine is always linked to thymine and cytosine is always linked to guanine. And one of the remarkable consequences of this structure, which was appreciated by Crick and Watson and was discussed in an article subsequent to the description of the structure itself, is that this amazing structure provides an explanation for one of the most mysterious questions in biology, which is the question of heredity. How is it that parents can convey information to construct offspring that resemble themselves? And the clue to this is right there in the structure of DNA, which Watson and Crick pointed out, that if the two strands are separated, then because of these rules of base pairing, it's straightforward in principle to construct new strands using the base pairing rules, so that adenine is always paired with thymine and cytosine is always paired with guanine. You can construct new strands that basically provide two replicas of the original molecule. So this is how a parent gives rise to two offspring, which contain the identical sequence of bases. So this, I think, most people regard as one of the most important and fundamental discoveries in biology. And this is the secret of heredity revealed at a molecular level. And much of the fascination about what's happening in biology can be traced back to this one discovery. Okay, so what's not always so appreciated, because it's not discussed so much, is that right there in the structure of DNA is a very, very fundamental mathematical problem. And this problem was pointed out to Jim Watson. So Watson was working with Crick in Cambridge, and when they stumbled across this discovery, he was terribly excited. He wrote to his mentor, Max Delbruck, who was at Caltech. And this is Max Delbruck, shown there in the photograph. And Delbruck was one of the pioneers of molecular biology. His name tends to be not so well remembered as that of Crick and Watson, but he was originally a physicist. He had worked with Niels Bohr and then had become drawn to biology and was the founder of a particular revolution in genetics concerned with the use of viruses, so-called phages. So he was a towering figure in the field at the time that Watson wrote to him. And what Delbruck immediately pointed out, writing back to Watson in Cambridge, is that the two strands of DNA are linked together. And by linked, we mean what's shown in the illustration on the right-hand side, which is two simple circles which are linked together in such a way that they cannot be pulled apart without breaking one of the links. Now, we have to be a little careful in trying to interpret this in the context of DNA, because DNA is a sort of linear strand. So the way I'd like you to think about this as we move forward in the talk is, whenever we think about DNA, we should always imagine the ends of the strands as being sort of tied together in a sort of imaginary loop, so that each strand forms a circle. Now, in practice, that's not the case, but what happens in practice is that DNA is a very long molecule, and it's believed that the ends of it are pretty much always fixed. In the case of bacterial cells, which just contain DNA, they're thought to be fixed to the membrane of the cell. And in the case of eukaryotic cells, which are the cells that comprise animals like us, they have a nucleus, and the chromosome ends are thought to be attached to the nuclear membrane. Either way, the ends can't move. So that's pretty much similar to thinking of it as looped in the way that I suggested. So what Delbruck is pointing out is that the two strands of DNA are linked in this way, and you can sort of appreciate that from the picture of the structure that we had on the previous slide. Okay, so what does this mean? Linking is a mathematical concept. So let's think about how we define linking mathematically. Okay, so let's begin by thinking of two circles which are linked in three-dimensional space. Now, we're going to imagine projecting this three-dimensional configuration onto a plane, as we did on the previous slide, in effect. And you see another example of it here. And we're going to choose a direction of movement around each of the circles. And it doesn't matter what direction you pick, it only has an influence on the signs of what we're going to do. So just pick directions arbitrarily. They're picked arbitrarily as shown on the picture. And you can see that whenever we do this projection, what we will end up with is a set of crossings where the two curves cross themselves in the plane. And there are two crossings illustrated for this particular link. And we're going to calculate a sign attached to each of these crossings. It's a very simple rule, but it depends on the fact that we've got these directions, these arrows. And the rule is, if the arrow that's on top has to be rotated clockwise to bring it into alignment with the arrow on the bottom, the sign is negative. But if the arrow on the top has to be rotated counterclockwise to bring it into orientation, then the sign is positive. And that's the rule that's shown there. Now, it's very important when you do this calculation that you only look at the crossings of one link over the other, because it's quite possible for one of the curves to cross over itself. So you imagine if you take the right-hand link and just twist it, that will create a crossing, but it will be a self-crossing of that curve with itself. So you don't want to include those crossings. You just want to include the crossings where one link crosses the other one. Now, the formula that we're going to calculate is very simple. You just count the number of positive crossings, you subtract the number of negative crossings, and you divide by 2. And the number you get is called the linking number of the link. And there's a very simple example that's shown at the bottom there. It looks rather complicated. It has 6 crossings. And if you run through what's happening in your head, if you look, say, at the top right-hand crossing, it looks like this. Is that right? No, it looks like, oops, it looks like that. So you have to watch your orientations here. And so when I move the top arrow coincident with the bottom arrow, I go in a counterclockwise direction, so I get a sign of plus. And if you run through the other crossings, you quickly get used to working out which direction the arrows are going, and it's a simple calculation, and you see there's 6 positive and 2 negative crossings, and so the linking number is 2. Now, there are a lot of crossings, but the linking number is actually quite low. And you can sort of see what's happening here, because if you look at the very top blue strand, you can see that you could move that strand upwards in such a way that it just comes away from the red curve, and the crossings disappear. And if you think about this, this doesn't change the linking number, because you lose a positive crossing and a negative crossing, and that cancels out. So this illustrates a very, very important part of the link about the linking number, which is fundamental to understanding it. It's an invariant of the topology of the link. And topology is the branch of mathematics that deals with the properties of mathematical objects which remain the same under continuous deformation. We can move things around, we can move one curve over the other, we can stretch them, we can imagine them as if they were made of elastic and we can stretch them out. We can do any kind of continuous transformation, provided we do not break anything. Breaking is not allowed. And so you could think of links being moved around in space. You can imagine any projection onto any plane of the crossing. And the theorem about this number that we calculated on the previous page, the linking number, is that it's a topological invariant. It doesn't depend on any of those continuous movements or any projection that we pick. It tells us something about the topology of the link. Now, the idea of topology really emerged early in the 19th century. The person who first defined the linking number was, in fact, the mathematician Carl Friedrich Gauss. You will know of him through the Gaussian distribution. He's often spoken of as the prince of mathematicians because he did so much in mathematics that was absolutely fundamental. And he had a habit of making amazing discoveries and just writing them down in his notebooks and not publishing them because he hadn't figured out everything about it. And so it was only after his death that this his formula for the linking number, he didn't calculate it the way we did on the last page. He did it in a very different way, but nevertheless, it's the same number. And he saw, as he noted down in his diary, that there was this new branch of mathematics that was concerned with the properties of things under continuous deformations. And the word topology wasn't coined at that time. It was, in fact, one of Gauss's students who coined the name. But it was called at the time analysis cetus, the geometria cetus, the geometry of place. And it was characterized by this fact that it wasn't dependent on the kind of geometric quantities that had been important previously, like distance or angle, or size or area, any of those quantities that are given by real numbers. Rather, it was dependent on these much apparently looser ideas like linking and crossing. And Gauss is saying in this little statement that very few people had realized that there was this new branch of mathematics, and we still know very little about it. And it was really only at the end of the 19th century that topology developed as a new mathematical subject, and it was extremely important in the development of 20th century mathematics. So what you're seeing here is one of the earliest and simplest topological invariants, the linking number of two circles linked together in space. Okay, so what does this mean for DNA? Coming back to Delbruck's point that he was making to Watson, well, it's easy to do this calculation now. So this is a little segment of a DNA double helix. It's basically one full turn of the helix. And you'll see that there are two crossings. And if you do the imaginary connecting everything into a loop, so there are two circles linked together here, and give it an arbitrary orientation, I've chosen one. What's the linking number? Well, let's see. The top strand is going this way. The bottom strand is going this way. If I need to rotate it that way, it's going counterclockwise. So that's a positive sign. And what about the other one? The top strand is going that way. The bottom strand is going that way. Again, I go counterclockwise. So it's another positive sign. So there are two positive and no negatives, so the linking number is +1. And if you look at the structure of DNA, to do a full turn of the helix requires more or less 10.5 base pairs. So of course, DNA molecules have hundreds of thousands of base pairs. And to know what their linking number is, we just divide more or less by 10. That's an awfully high linking number, and this is really the point that Delbruck was making to Watson. The two strands are linked. So how is it that if this mechanism that Crick and Watson suggested, the strands are separated and new molecules are constructed on them, how is that possible? How does the unlinking happen? It seems very mysterious. And at the time, this was considered to be a major objection against the structure of DNA. Sorry, we've just done that little calculation. The linking number here is a half of 2 minus 0, so we get 1. Okay. Right, so after Crick and Watson published their structure, so it's very funny, Crick, when he got Delbruck's letter, was actually extremely nervous and says that he spent a couple of sleepless nights wondering whether he and Crick had made a terrible mistake. But anyway, they thought they hadn't, and they published their famous paper. And the year later, Delbruck published a paper in which he discussed this problem of linking. And what he pointed out was there's basically only 3 ways you can think of to solve this problem of unlinking the strands. One is that you could try to slip one strand past the other. You could try unwinding one over the other. And he dismissed both of these as basically just implausible. They were too inefficient. Even at that time, people realized that these DNA strands were very long. And it just seemed very, very difficult to imagine that any process would be capable of doing that. And so he fastened on the third possibility, which is really the only thing you can do, because this is a mathematical problem, right? It's not to do with biology, it's not to do with physics. These strands are linked. Somehow, they have to get completely unlinked. The linking number has to go from something enormous to something which is zero. And he suggested a mechanism which is one of breaking the strands. You have to break the strands. You have to destroy the topology and then repair it. And he put forward a possible way of doing this, which is illustrated in the diagram on the right, which was quite an interesting idea at the time, but you see that it involves mixing up the two bits of each individual strand, shown by the crosses there. So this was the idea. It was a theoretical idea. Delbruck didn't have any evidence for it. And this was published in Red. And then subsequently, a very famous experiment was carried out, which convinced everyone that Crick and Watson were right and Delbruck was wrong. And this experiment is often regarded by molecular biologists as the most beautiful experiment in molecular biology because it was so compelling. Excuse me. It was carried out by Matthew Messelson and Franklin Stahl, also at Caltech. And we won't go into the details of it, but basically they utilized a very clever idea of labeling the parent DNA molecule with a heavy isotope of nitrogen, nitrogen-15, and then allowing the strands to be replicated as they are in a bacterium, an E. coli bacterium. And what they had was a new method of density gradient centrifugation, which is a way of being able to separate molecules which are just slightly different in weight. And by doing that, they could follow how this heavy isotope was moving in the new strands that were being constructed. And what they discovered was that Crick and Watson's original idea that the strands were being separated and new molecules were being constructed on each of the separate strands was exactly correct. It was the explanation that fitted the data exactly. The data, their famous data is shown there, but don't worry too much about it. It's just to illustrate how compelling this experiment was. So what they showed by this was that Delbruck's suggestion that the breaking and repairing was mixing up the pieces of the individual strands was wrong. The strands were remaining intact, and somehow they were being unlinked. So nobody understood how this unlinking was done, and Delbruck's other point that the only way to do this is by breaking was still a possibility. And this situation remained as it was for many years, for over a decade, until James Wang, who was at the time at Berkeley, but is shown here as a professor at Harvard, where he's now emeritus professor, did a remarkable analysis in which he found an enzyme that was capable of changing the linking number of the DNA. And he had an idea of the structure. It took many years and many other people's contributions before what was actually going on was understood. But these enzymes were christened by James Wang topoisomerases. And he called them, and I think quite rightly, cellular magicians. And they really are magicians. And what's shown at the bottom here is an illustration of how one of these topoisomerases works. This is actually not the kind of topoisomerase that James Wang first isolated. There are in fact two classes of topoisomerases, and we'll come back to that a bit later. This is the second class. It was discovered in the lab of Howard Nash, and how it works is that this topoisomerase enzyme grabs two double helices, the red and the green. It breaks the green double helix, and it pushes the red double helix through that break, and then it reseals the green double helix. And what you see is that this operation is not changing the molecules except insofar as they're placed in three-dimensional space. It's purely a topological operation, and it's altering the topology. And you might like to sort of think a little bit about by what is it changing the linking number? We'll come back to that in a moment. But this enzyme was so different from anything. Most enzymes sort of take a substrate and construct a completely new molecule, a substrate, add things to it, take things off. But this is just changing the topology. And as that quote from James Wang suggests, he had a hard time convincing his reviewers that this was real. But it is real. And since that time, these enzymes have been tremendously important in various aspects of biology. We'll come to that. So we've talked about the topology here in the context of the replication of DNA, of separating the strands, and the problem of linking. And the topoisomerases are now thought to be essential to that problem of linking, of unlinking the DNA. But I want to move now to a different aspect of DNA, which is the problem of transcription on DNA. I'm sure you'll be familiar with the fact that DNA encodes the structures of proteins that make up the sort of working machinery of the cells in our body. And the discoveries of molecular biology that came after Crick and Watson's discovery of the structure is that DNA makes RNA, which makes protein. RNA is another polymer molecule, ribonucleic acid, as opposed to deoxyribonucleic acid. And the information for constructing the protein is present in segments, so-called protein coding genes, on the DNA. In the humans have about 20,000 of these genes on our DNA. The process of converting the information on the coding region into RNA and then subsequently into protein is carried out by a very complicated machinery, some of which is shown at the bottom. There's a very interesting issue of how it is that genes are selected to be transcribed. Because if you think about it, all the cells in our body contain exactly the same DNA, more or less. And the thing that makes a difference to whether the cell is a skin cell or a nerve cell or a liver cell is which genes are being transcribed in that cell. So how is it that different genes are transcribed from the same genome? And the answer is that the genome contains not only instructions for the protein, it also contains sequences, so-called regulatory sequences, that instruct the cell as to which genes to turn on. And these sequences are bound by regulatory factors, proteins, which are called transcription factors. And it is that dictates whether in a particular cell type this gene is expressed or that gene is expressed. So this is the process of gene regulation, and it's very significant for understanding how the genome, the DNA, is interpreted. Okay, so the critical step here is how the coding region is transcribed into into RNA, I beg your pardon, and that's carried out by this enzyme called RNA polymerase, or RNAP, that's shown there. Now, I'm sure in your biology classes you'll have seen pictures of how this works, and you'll have seen pictures of RNA, RNA polymerase moving along the DNA and producing the messenger RNA. And I've just taken an illustration of this from Wikipedia, but this is pretty much what you will see in most textbooks. You have this linear motion around DNA, and there's something fundamentally wrong with this, and it's usually not discussed, but it's real. And the problem is that DNA is a double helix. And this polymerase has to track the bases on one of the strands. The only way it can do this is if the polymerase is rotating around the DNA. It's not just moving, it's not just translocating, it's actually rotating. Now, this rotation is almost never pictured in these depictions of gene expression, but it is real. And if you're a clever experimentalist, you can actually see it. And this is a nice experiment that was carried out by Kazuhiko Kinoshita's group. And you can see what's involved here. They've tethered the polymerase to the base of a microscope slide so that the DNA is free to move in the solution. They've tied a magnetic bead to the tip of it and used a magnet so it stretches out the DNA. And they've put fluorescent beads on the magnet. And so what happens when the RNA polymerase starts working is it's rotating, but of course it's immobilized. So what happens is that instead of the polymerase rotating, it's the DNA that rotates. And you can see that as the magnetic bead moves around. And those are the photographs showing this rotation is actually real. So for the most part, when you talk to biologists, they kind of gloss over this. Of course, they know it exists, but it's actually not so straightforward to think about it. And this is where topology again enters the picture. I think the first person to publicly, in the biological literature, sort of discuss the consequences of this is the Oxford biologist Peter Cook. And what he pointed out is that the RNA polymerase has to do two things. It has to move along the DNA, and it has to rotate. And there are basically four possibilities. Now, the two possibilities on the left, 1 and 3, involve rotation of the polymerase. So you can imagine the DNA is kind of kept fixed, and the polymerase is rotating around it. Now, this is fairly implausible, because the polymerase is a huge enzyme. It's like 80 proteins all coming together in a huge complex in humans. And the idea that this thing is actually rotating, there's so much inertia, the messenger RNA is coming out, and if the polymerase is rotating around there, messenger RNA would get tangled up or linked with the DNA. And we know of no way to unlink that. So this seems very implausible. The other possibility, illustrated on the right, is that it's not the polymerase that rotates around the DNA. Rather, the polymerase is fixed and the DNA is being rotated. Now, that seems more plausible, at least insofar as you can imagine what's going on at the level of individual molecules within the cell. And Cook's conclusion is that, and a conclusion that I think is by and large what most people believe, is that it's not the polymerase that rotates, but rather the DNA. There's some controversy and discussion about what's translocating here, and I don't want to really sort of get into that. I want to focus here on this question of rotation. And the inevitable consequences of the fact that the polymerase is being fixed and then it's winding around the DNA is that the DNA is being twisted. So what does that mean? Okay, so now at this point, I'm going to do something which is probably a bit silly. I'm going to try to do a little experiment. And you know, experiments never work when you do them in public because it's very bad juju. Okay, so I hope you can see that what I've got here is not DNA, but it's a rubber tube, a plastic tube, tied to a couple of handles. And it's very hard to sort of really replicate the way that a motor protein like a polymerase would work. But for the purposes of what we're going to do, it doesn't matter. What I'm going to do is to simply twist this by hand. Okay, so let's see if we can get this to work. I'm going to twist it, and you will see immediately that as I twist it, the axis of the tube is moving in three-dimensional space. And if I keep twisting it slowly, you can imagine what will happen. And what happens, and the experiment doesn't always work very well, is that the axis moves in three-dimensional space and begins coiling up. And if I carry on twisting, it ends up in this mess of coils. And this is the consequence of what happens when you start twisting a ribbon, a double helix, or a plastic tube. And you've probably seen this at work if you've ever had to play around with a garden hose. It's the same problem. And what we're seeing there is what is called in biology supercoiling. And supercoiling, let me just let that unravel, is a phenomenon that was actually detected very early on in the story of DNA by Jerome Winograd, who was at the time also at Caltech. Lots of things were happening at Caltech in the early days of molecular biology. And he was, in fact, the inventor of the density gradient centrifugation method that Messerson and Stahl used in their beautiful experiment. And here is an illustration. He's showing, I think, a telephone cord that is a good way to, in the old days, of showing supercoiling. But as you can see, supercoiling can result in a number of different kinds of shapes. And the shape in the middle, which is like one of the little coils that we made in our experiment, is called a plectoneme. That's a particular kind of supercoil. There are other sort of shapes that can emerge. And I should point out that what does emerge in terms of the shapes now begins to depend on the physics of the material. Right, we've talked about mathematics and we're going to concentrate on it. But ultimately, if you want to understand when a supercoil will form, where it will form, what shape it will have, you need to know something of the physics of the material, the bending, the torsional stiffness, all of those things. So that's going to be in the background of what we say here, but we're going to focus on the mathematics. So how do we understand this supercoiling from a mathematical point of view? Okay, now here things get a little bit more complicated. Okay, we have to think about the geometry. And what we're going to do is to imagine the double helix as a thin ribbon, and the edges of the ribbon are the two strands of DNA. Okay, so we think of it as a tiny ribbon. And we're going to mathematically analyze this by imagining that we have a parametric description of one of the edges of the ribbon, and it's described there by that function E, and S is a kind of parameter that tells us where we are along the curve. And we're going to look at this by thinking of a sort of coordinate frame along the curve. And the coordinate frame has a unit tangent vector, t, and a normal vector, a vector perpendicular to that, that's lying in the plane of the ribbon. That's u. Okay? So this is just how we set the problem up. And we're going to think about two kinds of behaviors that can happen to this ribbon in three-dimensional space. The first kind of ribbon behavior is the one in which basically the ribbon is basically just twisting along its axis, its central axis. And we can measure this by taking the normal vector u and asking how quickly is it rotating along the tangent vector, which I can't. My hands need to be sort of twisted to do this, but it's basically measuring that rotation rate. And the vector formula that we've got there, t cross u times u dot, dot product with u dot, is a mathematical way of calculating that rate at which that angle is changing. And of course, it's changing as we move along the, so we're going to integrate it along the curve. And then to normalize it, we'll divide by the total circumference of the curve, which we think of that as a sort of unit curve. We divide it by 2 pi. And this gives us a quantity called a twist. It's the total twist of the whole ribbon in three-dimensional space. Okay, so twisting is one thing that can happen, but as we saw in our experiment, it's not the only thing that can happen because the axis of the curve can also move in three-dimensional space. And to describe what happens to the axis, there's another quantity. It has a nice description. It's called the writhe. It's a lovely name. And now this formula looks much more complicated, but it actually has a simple interpretation. The way we try to measure how this curve moves in three-dimensional space is we take 2 points on the curve, and I've called them S and S primed. So it's the same curve E that we're looking at, but we're now looking at 2 points. And we look at the vector joining the 2 points, which is ES minus ES primed, and we look at the unit vector in that direction, which is dividing by the distance, the length of that. And what this gives us is a point in the unit sphere. And as we move S and S primed around the curve, this vector will move around the unit sphere, and it will create a sort of region on the unit sphere. And what we're calculating there is the area of the region. So it's a way of summarizing how, as S and S prime move and the curve is moving in three, it's summarizing the total amount of movement of the curve in three-dimensional space. It's actually a very clever way of summarizing that by calculating this area. And then it's normalized to the area of the unit sphere, which is 4 pi. And this gives us another quantity, writhe. Now, these quantities that we have calculated here, total writhe, total twist, are very different from the linking number that we calculated previously. They're real numbers. They matter how much you do. They're distances, if you like. But there's an amazing result that links these things together, and this is our beautiful theorem. It's the Caligiano-Weiss-Fuller theorem, and it says that magically, when you take a closed ribbon in three-dimensional space, if you calculate its twist and you calculate its writhe, these are real numbers, you add them together, and that must be the linking number of the two edge curves with each other. And this is amazing, because twist and writhe, as I said, are real numbers that depend on distance, angle, area. But when you add them together, you get something which doesn't depend on distance, angle, area. It's a topological invariant, depends only on the disposition of the curves in three-dimensional space. And instead of being a real number, it's an integer. Remember, we counted crossings. It's 1, 2, minus 1, minus 2. So somehow all the decimals disappear. And this is the basic formula for understanding how ribbons closed ribbons can be present, can be configured in three-dimensional space. Now, this kind of result, the links topology and geometry, is actually an incredibly important theme in 20th century mathematics. And this is a simple version, if you like, of much more powerful theorems. You may have heard of some of them: the Gauss-Bonnet-Chern theorem, the Riemann-Roch theorem, the Atiyah-Singer index theorem, which are theorems that relate topological invariance to geometric invariance. And this has been extremely important in 20th and 21st century mathematics. And if you like, what we're seeing here is a very simple example of this, which is about ribbons in three space rather than manifolds in any number of dimensions, which is what these other theorems tell us about. Okay. So we can have deduce something very quickly from the Caligiano-Weiss-Fuller theorem, which is that it provides us with an explanation. This is actually a sort of repeat of the little experiment that we did with the ribbon. It's with the tube. It's kind of showing you this interplay between link and writhe, and it's actually quite a nice illustration. So you could imagine these two dispositions of the same ribbon, one in which the ribbon has been stretched out and the axis of the ribbon is really lying in a plane. So if you think about what that means, it's actually straight. If you go back to the writhe formula and calculate the the distance, the unit vector between 2 points, that's actually giving you just a point. There's no area to it. So there's no writhe in this configuration. It's all twist. But then if you imagine the blocks at the ends of the ribbon being sort of brought close together and imagine this was sort of elastic, then I think it's easy to see that what's going to happen is that the ribbon's going to coil up in this way. And if you think about this, now there's no twist, or very little, because it's basically the ribbon is going like this, it's not going like this, but now it's writhing. And this is showing you how the linking number remains the same, but the twist and the writhe interchange. Okay, so it's a nice example, but it's really very similar to the experiment that we were doing previously. Okay, so as I was saying, the Caligurano-White and Fuller theorem gives us an explanation for something which biologists have discovered, which is that, as I mentioned, there are two classes of enzymes. And they're called type 1 enzymes and type 2 enzymes. Sorry, topoisomerase enzymes, the enzymes that change the supercoiling of the DNA. And the type 1 enzymes work by taking one of the strands of DNA and cutting it. And having cut it, they move the other strand through the cut, either through a sort of rotation or an actual movement, depending. There are two classes of enzymes. And then they reseal the strand. And then there's the type 2 enzymes. This is the kind of enzyme that I showed a picture of previously. This does something different. It takes the double strand of DNA, not the single strand of DNA, the double strand, and makes a double strand break and moves the other double helix through that strand. So there are two different kinds of operations. And if you think about it, those are exactly what you would expect from the formula. Because the formula tells us that if we want to alter the linking number, we can operate on two different things, the twist or the writhe. And if you think about it, the type 1 enzymes are operating on the twist, and the type 2 enzymes are operating on where the axis of the ribbon is in 3 space. They're operating on the writhe. And we can even calculate what the effect is on the DNA. So in the case of the type 1 enzymes, which use the twist, what happens when you take one of the strands of DNA, let's say the top strand, you break it, and you take the bottom strand through it, and you reseal it. And that's exactly what's shown there. And if you think about what happens, we put orientations on the curves, and it's very easy to see that what you're doing is you're switching from a positive to a negative crossing, or vice versa, depending on how the orientations are placed. And that tells you immediately what happens to the linking number formula. The link changes by plus or minus 1. So the type 1 enzymes are altering the topology by just 1 unit of linking number. Situation's more complicated with the type 2 enzymes, because the type 2 enzymes, we have to remember that we're thinking about the DNA double helix as a ribbon, and they're moving one piece of the ribbon through the other. So now the situation is as it's illustrated here, but now we have to be careful, because it looks like there are 4 points of crossing, but they're not. Because remember, for the formula, we have to focus on the crossings of one edge curve with the other, not with the self-crossings of the edge curve with itself. And then there are only 2 possibilities. It doesn't matter what they are. They depend on what's going on in the rest of the curve, so we just make any choices for the orientations and where the non-self-crossings are. And if you think about what happens here, when you do this movement, essentially the same thing happens. The sign of the crossing changes, but there are 2 crossings. So now, when you go back to the formula and decide what happens, the thing in the brackets is changing by 4, and when you divide by 2, it changes by 2, and therefore the linking number changes by 2. So we see here that the evolution has already discovered the Caligurano-White and Fuller theorem. It already knows and has figured out, a long time before we ever came on the scene, that to deal with this problem of linking, there are 2 solutions. You can change the twist, and if you do that, you change the linking number by 1, or you can change the writhe, and if you do that, you change the linking number by 2. Okay. So topology and genes are very closely linked here. All right, so I have just a couple of minutes left. So I'm going to be very quick with these last few slides. But I wanted to sort of point out that for a long time, even after supercoiling was discovered, the question of what the implications of it were for what's the working of the genome was really rather mysterious. And the reason for that was, although people believed it was going on, it was very hard to understand, to see it in action. And an important step forward was the model that was put forward by James Wang and one of his students about what actually happens when RNA polymerase is operating on the DNA. And this is called the twin supercoiled domain model. And now we're really back to the situation of DNA. We have to imagine that the DNA is fixed at the ends, which are those bars at the end, and we have polymerase, which is this motor protein that is translocating, but also it's twisting the DNA, right? And what Wang and his student proposed is that the consequences of it, well, it's going to form supercoils, we know that, but they're positive supercoils in front of the polymerase and negative supercoils behind. What is positive and negative? Well, what it means is that a positive supercoil is one where the linking number is greater than it would be if the DNA was completely relaxed. And a negative supercoil is one where the linking number is less than if the DNA was perfectly relaxed. And this twin supercoil domain model was just a hypothesis at the time it was put forward, but more and more evidence is accumulated for it, and it's broadly acknowledged to be what people think is going on when polymerase is working. Positive supercoils are being formed in front of the advancing polymerase, if it's advancing, and negative supercoils are being formed behind it. And in the intervening period since Liu and Wang put forward this model, a great deal of molecular knowledge has accumulated about how this process is controlled. There are topoisomerases that are involved in reducing the positive supercoiling and reducing the negative supercoiling. Now, as we've begun to be able to look at what's happening on DNA more, we've begun to realize that this phenomenon of supercoiling is really at the heart of what happens on DNA. And I can only just briefly go through a couple of important experiments that people have discovered. One, which was work from Sunny Xi's lab at Harvard, was the realization that it is the supercoiling of the DNA that is responsible for something that was quite mysterious when it was first seen, which is that transcription does not happen. It's not a regular process. We used to think that polymerase would arrive, elongate, produce an mRNA, another polymerase would arrive, elongate, produce another mRNA, and you would get more or less a sort of fairly steady, stochastic production of mRNA. That's not what happens. What happens is that polymerases seem to arrive in bursts, produce lots of mRNA, and then there's a pause, nothing happens. Another burst arrives. And so transcription is very bursty. And the reason for this is very mysterious. And what Sunny Xi's experiment showed and suggested is that certainly in the bacterial context, the reason for this burstiness is really the buildup of supercoiling, which has the effect of restraining the polymerase. The DNA gets so supercoiled, the topoisomerases are unable to relax it fast enough, and polymerase gets halted. And that inevitably leads to this burstiness of transcription. So we're beginning to understand that this supercoiling has really important effects on how genes are expressed. It also has other effects, and these are experiments that were carried out by Cees Dekker at the University of Utrecht. And what he observed, again, with very clever experiments, single molecule experiments, is that these supercoils that are formed on DNA are not static entities. And it's very easy to imagine that they form and that's it. Actually not, because as forces are exerted on DNA, these plectinemes can move, they can diffuse along the DNA. And what's even more impressive, they can hop. A plectineme can very quickly, in milliseconds, disappear, convert into twist, and then reappear as writhe in another plectineme a long way away, thousands of bases away. And not only are these plectinemes moving around and hopping, there are proteins, shown here, a protein called condensin, which can actually collect them together and form giant plectins. So what's emerging from this is that DNA, through the problem of supercoiling and the action of topoisomerases, is having a very dynamic structure. And this dynamic structure is really at the heart of understanding how DNA is laid out in the nucleus. And I'm sorry, I can't sort of get into this too much because we're a bit short of time. But let me just say that this problem of what's happening, the consequences of supercoiling, are really at the heart of our current mathematical challenges in understanding how DNA is working. We have a pretty good understanding of it in the context of individual genes, which we think of in a fairly sort of discrete way in terms of patterns of proteins on DNA. But to accommodate supercoiling, we have to think about shape, the shape of the supercoils, where they're forming, how they interfere and interact with the proteins on the DNA. And this is a really formidable challenge, and we are still, we're only at the start of trying to grapple with this problem. And then finally, I will leave you with the fact that something that I've left unsaid is that it's actually very dangerous to break DNA, because DNA is the hereditary material, and breaking DNA always opens the possibility of creating mutations. And we can exploit that fact for drugs, but it's also something which evolution has strangely adopted itself. It uses breaks to make regulation. And this is potentially something very dangerous. And it illustrates the fact that evolution doesn't look forward to think about whether this is a good idea or not. And these have very important implications for certain kinds of diseases, neurodegenerative diseases, for instance. So I'm sorry to have rushed at the end, but we are getting really short of time. But I just want to leave you with an idea which perhaps you may not have realized from reading biology and hearing biology as it's spoken, that right back to the foundation of molecular biology, right back to the structure of DNA, the question of topology and the implications of topology have been absolutely fundamental to understanding how the genome works. So topology lies at the heart of biology. So my apologies for running over time, but thank you very much for your attention.
Okay. Hi, Grenaldera. It was a really excellent talk. In the YouTube channel, there are really people say that it's really awesome talks. I really didn't know about this connection between topology and genes. Okay, so there are some couple questions we got so far. Number one is, do TW and WR have an influence on the protein folding too? Do you think? Protein folding?
Okay, so that's a very interesting question. So proteins are also linear polymers, and of course of amino acids rather than of bases. And so these amino acid chains are also arranged in three-dimensional space. I think the topology of that is important. And one of the very surprising discoveries of in biology some years ago was that we used to think of these amino acid chains as just sort of folded up into sort of either sort of loose arrangements or sort of more like globular clusters. But it turns out there are examples where this single chain is actually linked with itself into a knot, so that protein molecules can actually be knotted. So it's not so much the twist and the ride that become important, I think, in the context of the protein molecules, but topology remains very important because proteins can be knotted, and this knotting is sometimes very important for the function of the proteins. So this is not something that I've been able to discuss in this talk, but I think the question was a very important one because it reminds us that it's not just the DNA molecule, which is the sequence of bases, that is afflicted, as it were, by the problem of topology, and particularly through the double-strand nature of the double helix. But the protein molecule, which is, of course, a single strand, it's not a double strand, itself has a topological affliction, which is the formation of knots.
Yeah.
So not so much the twist and right, but certainly the topology.
Yeah, so actually someone asked a related question. Why double helix? Because it looks like very inefficient structure because, right, compared to the protein, right? So one asked the question, why double helix? Why cell love these complex systems?
Well, it's complex at one level, but it's, I think, also beautifully simple at another level.
Okay.
Because think about how to solve the problem of heredity, right? One has to convey an amazing amount of complicated information, you know, how to make all these proteins, how to express them in the right place in the right cell. And evolution has found this astonishing way to encode this information on a sequence of bases, of 4 bases. But so that's an amazing thing. But then it has to solve the problem: how do I convey that information to a daughter cell?
Right. Okay.
And so the solution of having base pairing and these intertwined helices provide, I think, a really beautiful solution to that problem, because it allows the strands to be separated with all the complexity involved in that, and then the base pairing allows the molecule to be reconstructed.
Okay.
So it allows us to convert the same molecule, a double-stranded DNA, into 2 of the same molecules, double-stranded DNA. That's the problem of heredity.
Okay.
So I think that is a very simple and beautiful solution. I completely agree, however, with the statement that the consequences of this beautiful and simple solution is then to introduce a huge amount of complexity in another space. And the complexity involves the fact that these strands, as Delbruck pointed out, are linked together. And that has ramifications which are, you know, become central to much of the rest of biology, as we've discussed.
Cool.
So I think it's an illustration of the fact that evolution is dumb. It doesn't think ahead. It doesn't think, oh, that's a clever solution, but this is going to have really bad consequences down the road, so maybe we should try something else. It just does what looks of it'll work, and then has to deal with the consequences of that. So we get this combination of simplicity in one dimension and great complexity in the other.
Yes, great. And then there are actually several more coming. So another question comes from Yunmin Song. He asked, in eukaryotes, DNA is winding around the histone proteins. Do you think the histone protein also contribute to the linking number or supercoiling?
Absolutely. And I should apologize to those who know more about DNA here.
Sorry, he's my student.
The question's absolutely right. In the context of eukaryotic cells, the cells that make up animals like us, as opposed to bacteria, DNA is not free, but is actually wound around certain protein cores that are called nucleosomes. Now, the moment I made the statement wound around, it's telling you that the DNA is moving in 3-dimensional space and is writhing, and therefore this absolutely has an impact on the overall linking number and the topology. And to really understand what's happening in the eukaryotic setting is much more complicated because of the presence of these nucleosomes around which the DNA is wound. It fundamentally alters the topological issues. It's very different in the bacterial setting, where the DNA is not wound around these cores. There are proteins that link with the DNA, and they're important, but there are no nucleosomes. So it's mostly bare DNA. And a lot of the experiments that I described, plectinine hopping, the transcriptional bursting experiment are carried out on bacterial DNA, which is free of nucleosomes.
Okay.
And a very important challenge in the field is really understanding what happens when we move to the much more complicated context of eukaryotic DNA and nucleosomes. So there's even it's been a long-standing question of what the effect of even a single nucleosome is on the linking number of the DNA. It's sometimes called the linking number paradox, and I haven't had the time to get into those issues.
Okay.
But the question is right on the ball, because the moment you want to think about the topology, the nucleosomes play a huge role.
Why don't you give one more talk next time about that?
Yeah, well, that would be another talk altogether.
Yeah. And another question from Hyokpyo Hong. Thanks for the wonderful talk. My question is whether the transcriptional bursting due to the supercoiling is beneficial for regulating gene expression or not.
Sorry, I didn't hear the last part of that.
The transcriptional bursting due to supercoiling is, beneficial for the regulation of the gene expression.
Whether it's actually used to coordinate regulation of the genes. So I think the answer to that must be yes, because I think evolution always takes advantage of whatever situation it has at hand to exploit, as we saw with the question of the linking of the DNA strands. But I think the way in which this bursting is used to orchestrate how genes are expressed still is mysterious and isn't sort of completely understood. It could well be that the bursting is sort of helpful because it makes it easier, for instance, to sort of allocate resources across the genome. Because if you think about it, there are only certain number of polymerases that are present on the genome. And there are a lot of genes. Lots of things have to be expressed. So how do you allocate? How has evolution figured out how to allocate? Sort of polymerases. And you could say, oh, well, we're going to sort of focus on this gene because it's important and we're just going to sort of express it. But then none of the other genes might get left out. And maybe the supercoiling and the consequences of bursting have been exploited to sort of allow a more equitable kind of solution in which sort of polymerases have the ability to sort of come and do a burst here, come and do a burst there, and sort of spread their effects across. I'm guessing. That's a speculation. Nobody's sort of developed the theory or followed through the experiments to sort of confirm something like that. But one could imagine that would be a way in which the transcriptional bursting is exploited.
Okay, got it. And then another question is from Seo Park. Actually, there are various types of the supercoils. Do you think it's possible to find the type of the topoisomerase which is the most efficient to annul the specific supercoiled DNA? Kind of, there are type 1, 2, and how can cell know what is the best one to choose at this point?
Yeah, that's a very interesting question. And I suspect there may well be forms of regulation like that. It is known that the different topoisomerases, and I should say that there are two classes of topoisomerases and subclasses, but there are often many examples within, say, the human genome of topoisomerases within those subclasses. So there's lots of different kinds of those topoisomerases, and they do different things. And although that story hasn't been completely worked out by any means, it is known that certain topoisomerases tend to find and resolve certain kinds of topological problems, certain kinds of supercoils.
Okay.
And I was mentioning the fact that there's a protein called condensin, which one effect it has is to do plectineme absorption. It sort of collects together plectinemes into a single plectineme. And part of what condensing does is to extrude DNA into loops. And while it's doing this extrusion, it's doing this plectinine absorption. And it's known there are particular topoisomerases that are attracted to those sites where condensing is working and work to resolve the supercoiling around that. The question of whether the different types of supercoiled structures, we've talked about plectinines, but the question is quite right that there are many other kinds of supercoiled structures. Whether they're discriminating enough to tell apart those and work on specific things is not known, but I could imagine that there is some ability to sense that. The suspicion, I think, the feeling that most people have in the field is because you have to ask, how does the enzyme know what kind of plectinine it's dealing with, right? You can't see it. It's probably the case that the topoisomerases are sensitive to mechanical strain, to the torsional strain that's present on DNA. And that may be what is as driving them to certain sites in the DNA. Whether that's enough for them to discriminate between different structures of supercoiling, I think is much harder to tell, and certainly we don't know that.
Gotcha. Okay. So, in fact, there are actually several more questions, but unfortunately, due to the other schedule, we have to close your talk now. So, thanks so much. Actually, thanks so much to show the beauty of the topology inside of our cell today. Actually, that means a lot for our Math Society and all our public participants today. So thanks so much, Dr. Guenardera. So let's thanks everyone to Dr. Guenardera's talk.
Thank you very much, Jay. It's been a pleasure, and thank you to all of those who participated. I can't, unfortunately, see you all, but thank you very much for your attention. I appreciate it.
Hopefully you can visit Korea and give a in-person lecture in near future.
I would love to do that, and I'm very sorry that I was unable to be there in person this time.
It was really perfectly fine this time. So okay. So 오늘 이렇게 참여해 주셔서 모두 감사드리고요. 이상으로 대중강연은 마치도록 하겠습니다. 긴 시간 함께 해주셔서 오늘 감사드립니다. 네. Yeah, Dr. Guenardera, now we are closing the Zoom and YouTube soon. Okay, let's keep in touch.
Thank you very much.
Bye-bye.
Bye-bye.